I'll be giving this talk later this week at the Northwest Science Conference, so some of the slides are intended for that audience. The only reason I mention that now is later, if anyone has any comments that would improve the talk, I'd appreciate it. So, as a little bit of background, several years ago we started a project with white pine that isn't related at all to what I'm going to talk about here, but it is where we came up with the question. We did a bunch of foliar nitrate or foliar nutrient sampling in white pines at the Swan property, and found that total N and foliar nitrate varied between lots of the different trees there, and the higher nitrate seedlings were more heavily browsed by deer and were therefore shorter. There were lots of other nutrient differences too, but the project that I'm going into talks more about nitrogen. This got me thinking about nitrogen relations in a nutrient cycle as applies to white pine forestry. And then also, I've looked at where many hundreds of these white pine seedlings have been planted at North into this fragmented landscape that I was talking about yesterday. And the duff layer is extremely variable among these planted sites. Some places completely absent, some places it's a deep, nice organic horizon. And so I started reading about what impact that could have. And of course, we know Morgan mentioned yesterday, plants do use some simple organic end molecules, and so that a contemporary nitrogen cycle plant use diagram now, plants and microbes both directly compete for some amino acids in some systems. And then, so there's quite a large body of research accumulating on this. Amino nitrogen pools are large and ecologically relevant in some systems, particularly forest systems with duff layers and lots of recalcitrant, slow decomposing sources. And many plant species will directly take up amino acids without them being mineralized by microbes. And this is reviewed pretty well in New Phytologist 2009. So I wanted to know if this is relevant for white pines. And so just a simple experiment to kind of start learning about this. And this is one of our seedlings up there that at last year was about 5 feet tall. So they're growing well. So again, continuing to sort of think about how this could matter for white pine forestry, we know that harvest and other major disturbances in forest ecosystems can change the relative availability of soil and species. After clear cutting, for example, there can be a lot more nitrification, so you can have more nitrate in soils. And conifer seedlings often struggle. Very few conifers take up much nitrate. It's not a very valuable nutrient for conifers. They prefer ammonium. And there's little, even if you try to make them grow on nitrate, they typically don't do very well on it. And again, there's from the 80s and then into the 90s, a lot of research on that, and particularly with trying to get replanted conifer species into clear cuts and having those plantings struggle. Nature papers, Vidisek in Ecological Monographs kind of was an early paper on that. So finally, what white pines do we have on site at the Swan property? We have a number of regenerated trees. There are old, tall white pines producing a lot of seed. At various times they've germinated and grow. So we have what I'm calling the wild type or Swan Valley type trees. We've also, as I mentioned, planted several hundred rust-resistant hybrids. I don't really have time to talk about the hybrids so much, but there's blister rust is a disease that's wiped out many white pine populations in the Pacific Northwest. In the 1960s, researchers in Idaho started developing a rust-resistant cultivar from seeds collected from trees that didn't get the disease. It's called the Bingham genotype cultivar, was developed from northern Idaho populations of trees. We know that there weren't any Montana trees selected for that resistance development. And so, northern Idaho is real classic white pine habitat, deep, fairly well-drained soils. It's not as harsh of a winter and a lot more water. In contrast, the Swan Valley is really cold, tight clay soils, shorter growing season, potentially could select for some different traits in the white pines if they were there long enough. So, first question you might think about is that, they took a small number of seeds from these sites in Idaho and developed a cultivar. Would that make a genetic bottleneck for the species? Kim et al. In 2003 answered that no. Heterozygosity was really high in these cultivars. But we learned in Trends in Ecology and Evolution in 2003 that even if you have robust genetics in your cultivar stock, they might not contain traits that are really good for your spot where you want them to go. So that last slide might have been throwing a little bit of a curveball, but putting all this stuff together, with a couple of questions I wanted to start looking at. And one, does amino acid uptake occur with white pine? It's a simple question about white pine biology. Two, does genotype matter as regards nutrient uptake? The swan is a much more difficult place to grow, colder. Possibly there's a reason that amino acid uptake would be relevant there. And then finally, a couple of papers in plant nutrition research have wondered about what happens to trees when they are not given ammonium. Do they compensate by using other resources? We know it's not nitrate, maybe it's amino acids. Methods. We grew some seedlings at the University of Montana. We grew 200 seedlings. 100 of the rust-resistant, or BG, is what you'll see the rest of this, or in the wild-type Swan Valley genotype. I grew them for a year. That's Emily Stark. She managed the greenhouse and was really helpful at the forestry department at U of M. And we trained these seedlings in certain conditions for the whole year they were grown there. And so this diagram just kind of tries to show that. On the left, the Bingham genotype, the right, the Swan Valleys. All of these were given standard micronutrient greenhouse mix. And then they were all given arginine, glycine, and nitrate in close to field relevant concentrations, and then balanced so they were getting the same molarity of nitrogen. And then half of them were given ammonium in that mix and half of them were not. So that's how they were trained for a year. Then after that year, we prepared for a hydroponic laboratory experiment. We rinsed the soil off the roots. There's one that I've just pre-rinsed. And there was a control sample. These were, as far as we could tell visually, these weren't colonized by mycorrhizal fungi. So looking at genotype times which of the nutrients in question, which of the end sources, arginine, glycine, nitrate, or ammonium, and then whether or not they were trained on ammonium times the controls made 38 different treatment combinations. So I don't know if I can adequately do that in the amount of time I have, but I'll try and describe the methods as well as I can. I really closely followed the methods of Oland and Nashholm in 2004, Tree Physiology, and I can provide that reference. So recall this diagram, and then I make a schematic out of that and try to get at what the sample groups were. So we have the two genotypes, the ammonium training, and then I took subgroups out of each of those 4 sets and put them in the incubation. Where was let me see if I have a picture out of order. Maybe I don't. It would be, that. This is what the incubation looked like, the hydroponic incubation. So this is seedlings in falcon tubes on a shaker table. So how did I choose which seedlings went into those groups? And that's what this whole thing is. So the seedlings, took them out of the soil, rinsed them, put them in the falcon tubes, and they were given the exact same nutrient mix that they had been trained on, except that in each subgroup, the arginine, or the nutrient of interest, was replaced with one labeled with heavy isotope, C or N. C and N in the case of arginine, and N in all the rest of them. So again, each subgroup of seedlings that I pulled out got all the same nutrients that it got before. It's just one of them was labeled. And what that was going to allow me to do later is to say how much arginine was the tree able to take up, how much ammonium was it able to take up. So I had to put a separate label into each group to learn that. The dual labeled arginine, reason for that will become clear in a second. So the training groups, they got the same things as they were trained on, except I took a subgroup out of the ones that had never received ammonium in the greenhouse and then gave them ammonium just for that 1 hour of incubation. So what that would help me learn is if these seedlings were starved of ammonium for a year, they never got that nutrient, or I don't know that they got none. There could have been mineralization in the greenhouse, but probably very little. And if they suddenly have ammonium available, are they able to take it up right away? So finally, to learn that this was actually active uptake occurring, I nested an unreplicated seedling from each nutrient group and included a cyanide-producing compound in the solution that blocks active uptake by roots, because some of these nutrient ions will leak across the root membrane. And so what I was interested in is, do the white pine roots actually transport amino acid molecules through membrane-bound transporters? So I wanted to be able to eliminate leakage from that. So again, a lot of categories to look at, but it gets a little simpler in terms of looking at the data, because this ends up turning into just 3 main experimental groups. So we have the R to R group is where they were restricted. They were never given ammonium in the greenhouse, and then in the incubation treatment, they still were not given ammonium. They were only given the other 3 end sources: arginine, glycine, and nitrate. Then we have the restricted but then given ammonium, so the restricted to all, the R to A group. They were then given ammonium in the hydroponic assay. And then finally, seedlings that were given all 4 sources of nitrogen for the year in the greenhouse and then incubated with all 4 sources. And in groups, so I'm just showing that the groups where active uptake was necessary here. Like I said, there's 38 combined treatment groups here. The passive groups were all unreplicated. They're all of N of 1. Started out with 200 seedlings, had some attrition, and then 38 total groups. So kind of hard to maintain big N in each individual group. Then finally, there's a calcium chloride rinse that rinses the ions off the roots so you don't continue to have uptake. We separated roots from shoots, recorded mass and length, dried them, and then sent them to NAU, where Melanie Caron was really helpful in helping me learn about isotope methods and so forth. Finally, modeling approach in R. Some results. So several questions here. I'll try and go through in the order I presented them. First, we did recover excess label from the amino-enriched seedlings. So those subgroups of seedlings where we put an isotope labeled arginine and glycine in, we found that isotope enriched in those seedlings. So we conclude from that that they do take up arginine and glycine. And I'll mention here, and I'll come back to this in a few slides, the data I'll show is all from roots. We separated the roots and shoots, and the signal was much stronger in roots, and in the discussion I'll kind of mention why that is. So this is in the roots, and then in terms of looking at this chart, this micromoles of 15 N per gram, so this is the heavy isotope of nitrogen used, and that's how much micromoles of the heavy nitrogen per gram of plant tissue was recovered in the mass spectrometer, in excess of background levels. So we took some white pine seedlings that were never given any isotope and also ran them through the mass spectrometer and got a base level of how much 15N would exist in them naturally. So this is all in excess of what would naturally occur. Passive uptake estimated from those unreplicated samples. So we do confirm with this that there's active uptake of arginine and glycine occurring in western white pine. They have that ability. Then we also use, this is why we use that dual labeled. So the arginine we use had isotope labeled C and isotope labeled N, because there's always the possibility that there could be a soil microbe that would, or an extracellular plant enzyme that would break the amino acid first and then mineralize it, and then the plant would take up mineral nitrogen and you would erroneously conclude that it took up the amino acid. So in arginine, the C to N ratio in arginine is 3:2. So what we look for is that we recover that same ratio of C to N in the seedlings that we measure. And with a moderate R-squared value, and a slope of 0.68, 3:2, the slope should be 0.66. Pretty reasonable to conclude that we had intact uptake of arginine through membrane-bound transporters. Second question, how are they using these isotopes? Recovered N varied based on which isotope. We would expect low nitrate. We didn't know what to think about the glycine and arginine. And I'll get to more detail about that in a second. Genotype emerged as insignificant in here, and I made a mistake. If you read the abstract where I talk about how the amino acid uptake was different between genotypes, I made the kind of overexcited assumption when I saw this genotype times isotope interaction, and then I started looking at interaction plots and so forth and saw that the Swan Valley seedlings their uptake of the amino acids was in a different direction than the Bingham genotypes. But that was really because of this strong effect of isotope. When I worked with the data later, the genotype interaction was not a story. There wasn't any real relevant reason to suspect that the two genotypes were really behaving differently. So that interaction is a little bit of an artifact. The primary goal of looking for amino acid uptake we did find, and then this is kind of what got me a little more excited, is that we still found this compensatory uptake of glycine. There just wasn't a difference between the genotypes in this. So kind of trying to walk through this multi-panel plot here, we have the 4 nitrogen sources of interest: arginine, glycine, ammonium, and nitrate. And then the y-axis is the same for all of them. And again, this is how much 15N we recovered. But at this point, all this is a proxy for how much of the nutrient that the seedlings took up. So we can see here that the seedlings took up quite a bit of arginine compared to ammonium, which is the next most in uptake, then next glycine, and a very small amount of nitrate uptake. So that's the first thing in this multi-panel plot that I notice. Nitrate, so again, here's our treatment groups. All to all, so the AA are the ones that received ammonium the whole time they were in the greenhouse and then continued to receive it in incubation. R to A, they did not receive any ammonium, and then they were given it in that last hour of incubation. And then R to R, they never received ammonium ever. So this R to R group, even though they weren't given any ammonium, they didn't change their nitrate uptake. They didn't, try to access that as an end source. It just seems to be very low constitutive ability of this conifer species to do anything with nitrate, or very much, which is consistent with other research on conifers. Ammonium, this is a non-significant result that is meaningful. The ones that were given ammonium for a year and then continue to receive it, we recovered that much ammonium. The ones that weren't given any ammonium for a year and then only had it for 1 hour took up about the same amount. No real difference between the two. So it didn't take much effort for the white pines to be able to start moving a lot of ammonium in. I should mention at this point that active uptake occurred for all of these N species. I only showed it with the aminos earlier because I think it should be pretty obvious that active uptake occurs with with ammonium and nitrate in plants. Finally, with the glycine here, those were given ammonium for a year and then continue to receive it. That's kind of a control level of glycine that the plants are able to uptake given that suite of N. Those that were restricted of ammonium and then given ammonium, it didn't change. It seemed, what I would conclude from this is that once the ammonium was available, they were much more likely to start taking that up. But when seedlings were starved of ammonium for a year and then not given any ammonium in the hydroponic assay, they significantly increased glycine uptake. So it looked like an N source that they could use in absence of ammonium. And then finally with arginine, I don't have a lot of great explanations for what happened here. Those that were given ammonium and continue to receive it, this is sort of a baseline level of arginine that they were taking up, just assumedly because it's available. Those that were restricted from ammonium, but then given ammonium, the arginine uptake significantly dropped. And possibly the new availability of ammonium caused the seedlings to really want to go after it and increase transport of ammonium. That's possible. I don't really know why. And then also tremendous extra variability with the restricted group. And I guess the one more thing that I notice about this is that the arginine uptake level is so high, even compared to ammonium. And this is consistent with previous research that's looked at a similar assay on this in conifers as well. One explanation for this is simply that there are more nitrogen molecules available per molecule of arginine that gets transported across. There's 4 per molecule, and there's only 1 per molecule with ammonium. So every time the plant burns a proton to get a molecule, it gets more nitrogen out of the arginine. It's maybe one explanation for it. So kind of a lot to take in with these results, for sure. And then a couple of kind of last comments in terms of discussion from this, that other things I learned from this study that I think are pretty relevant that aren't direct data. One is that the root tissue separation, as I kind of mentioned earlier, turned out to be really important in this case. I mentioned the Nashholm paper from 2009. I actually emailed with him quite a bit before doing this. And one thing we discussed is that in these assays, researchers generally tend to just send the whole plant to the mass spec facility. They'll grind the whole plant and then sample from it. But that's kind of a poor idea if you think about it, if you want to learn about end nutrition in plants. Once a complex molecule like an amino acid gets into the root, the plant might split that molecule and send the nitrogen one place and store the carbon someplace else. So that C to N ratio might not show up. You can also have fractionation during nutrient assimilation. I don't want to spend a lot of time talking about that, but you can end up reducing the amount of your recovered isotope label unless you get it really quickly. So the 1 hour of incubation and then taking just roots and sampling them seem to work. I did analyze the shoot data and saw no significant relationships of any kind and very low isotope recovery. So roots seem to matter. And that might also just be translocation speed. The plant doesn't move a nutrient immediately. And then kind of relevant contributions from this study. It's the first that I know of observation of compensatory amino acid uptake during ammonium starvation. But this is a field, this is a hydroponic assay. You soak plant roots in a nutrient solution that doesn't at all mimic behavior that would happen in soil with competing plants and soil microbes. And there are some amino uptake field studies that have occurred, but very few. And so this is a field that desperately needs field studies to show some ecological relevance or determine if there is any. And I'd like to do that. Parting here with a picture of a really sad, overbrowsed white pine that had really high foliar nitrate. And kind of led me to ask this question. Like many here, thanks very much to the Gurnis family and the College of Forestry and Conservation for growing these seedlings and doing a good job taking care of them, NAU for the isotope work, and then folks in the lab and a lot of advice that I got from other MPGers. So thank you very much.